Good morning, everyone. Thanks to IJCNN 2025 for organizing such a conference, and thanks to all the committee member and the sessions here. Thanks to all of my co-author and my supervisor, Professor Kerry Fakri. I am SM Taslimuddin Raju. Today, I am presenting our accepted papers. The title of the paper, GNN-ViTCap, GNN-Enhanced Multiple Instance Learning with Visions Transformer for Whole Slide Image Classification and Captionings. And here is our presentation outlines. Here is our introductions. The first background, histopathology is a microscopic examination of the tissues. This is normally served as for cancer diagnosis and the treatment decisions. And this process involves the pathologist using a microscopic to evaluate the tissue samples for morphological or irregularities to provide the essential information for clinical decisions making. And histopathology can be divided either patch-wise or the slide-wise and analyzes based on this way. And the histopathological caption that are extracted from the diagnosis report that provided by medical authorities, and then this report are paired with the image patches. Therefore, automated generation of the diagnosis captions from the whole slide image could reduce the pathologist's workload. But the problem is here. The problem is treatments. The whole slide images are larger in size, each whole slide image 4 GB or the 5 GB, which make it difficult for computational analysis. On the other hand, multiple instance learning that normally uses for large images that divided the whole slide images into thousands of independent patches for feature extraction and the classification, but it neglects the vital tissue context and the spatial interactions. On the other hand, whole slide image can be divided into two ways. One is the scalar whole slide image, another is the microscopic whole slide image. In the scalar whole slide image, the absolute position is known. On the other hand, microscopic whole slide image, the absolute position is unknown. But microscopic whole slide image saves the cost and memory, but the problem is here, it will generate here the lack of the position, and the another one, it will generate the redundancy patches due to the subjective capture of pathologists. If the pathologist thinks that there is a high chance of the captions for particular reasons, then pathologists extract the more patches from these reasons. So it will increases the redundant patches. So we try to solve these problems. On the other hand, the sequential model like the RNN, recurrence neural network, and LSTM face the vanishing gradients problems that limit the long-range dependency capture in pathological data. Here I will describe some previous works. The first one, multiple instance learning divided into the two way. The one is the aggregate attention-based aggregation, and the second one is the graph-based aggregation. Here I describe the most four famous multiple instance learning method that use the attention-based aggregation. The first one, attention-based multiple instance learning, they use the learnable neural network to enhance the contribution of each feature. Similarly, the second one, they use the dual streams multiple instance learning, the transformer based multiple instance learning, and the last one is double tier feature distillating multiple instance learning. All of the method use the multiple instance learning, but they neglect the spatial relation among the patches. On the other hand, graph-based aggregation studies, for example, the first one, Deep Graph Shubra, they developed a graph convolutional neural network and they combined the local features with the global topological information, and they try to solve the problem of spatial relations. The other method like DSML, GDMLs, they also use the graph neural networks. Here, I will explain the most recent work that used for caption generation. The first one, Sunuki et al. They are the founder for caption generation models by using the histopathological whole slide image. And they use the two feature, pretense feature extractor, EfficiencyNet, and the DanceNet. And after extracting the features, they use the RNN-based decoder for caption generation based on the extracted features. On the other hand, other two authors, the Queen et al., they also use the same dataset for caption generations. But still, they did not care about the redundancy of the features. We try to solve this one. So this is our contributions. We proposed a GNN-ViTCap architecture for a continuous classification and a caption generation. And this architecture consists of visual feature extractor and the deep embedding clustering and the graph-based aggregations, and finally use the large language model for caption generations. The deep embedding clustering applied for remove the redundancy of the patches, and the graph neural network used for aggregate the patches relations, And then finally, visuals feature aggregate into the large language model, the combining the visuals feature and the textuals features to generate the captions. And in our studies, we use the two dataset. The one is the fetch text dataset. This is used for caption generation, and the breakage dataset. This is used for classifications. And this is our proposed architecture. We divided our proposed architecture into multiple modules. The first one is the fetch extractions. So in here, the pathologists extract the patches from the whole slide images, but we don't know the position, absolute position of each patches, and there are also the redundancy of the patches. In our second module, this is the feature extraction module, we use the Bayesian transformer and the ResNet50 to extract the features from here. After extracting the feature, now we will remove the redundancy from our features, and for that we use the deep embedding clusterings. In the deep embedding clustering, we first initialize the random k number of the cluster, and we randomly assign the patches among the clusters, and then we calculate we use the t student distribution. So in the t distribution, it assigned the soft probability. The probability determined if images or the patches I will assign to a particular cluster kk. Then we repeated this process until the divergence. We use the KL divergence in this case. So when our clustering done, then we will find similar images or the redundancy patches are contained into the same cluster. After that, we will extract the most represented features from each cluster, and for that we use the attention-based mechanisms. So in the attention mechanisms, we divide it into the query, key, and the value, and then we calculate the weight for each passes among each cluster. After that, we select the highest weighted images or the features from each cluster. After then it will remove our redundancy from the images. Then we designed the graph. Here we don't know the spatial relationship among the patches, or also the absolute position is unknown. So we can design the graph to establish the relationship among the patches. So we can consider each node as a features, and then we calculate the quotient similarity between the patches. And after calculating the quotient similarity, we pass these values to the soft global functions. So it will assign 1 or 0. If there is a 1, that means we can say that there is a relationship between the node I to the node j. Otherwise, there is no relationship. After our graph constructions, then we use the graph attention mechanisms to aggregate all the features. Therefore, we pass these features to the fit for all neural networks, then it will calculate the probability either it is the malignant or malignant. So this is our classification tags. On the other hand, we also use them captioning stacks, but in the captionings, the large language models are not directly compatible with the visuals features. Then we use the projections layer here. The projections layer convert the visuals features into the another dimension that is compatible for the large language model. Then we first the encoder. In the encoder, we first the visuals features. In the decoder, we first the true captions, and we fine tuning the large language model for caption generations. And first, we will explain our theoretical aspect of our proposed method. The multiple instance learning, it divided the whole slide images into multiple patches, and then it will assign the 1 or 0 based on the classifications, and then it aggregates it and then takes the final decisions. The visuals encoder, we use the ResNet15s and the BIT, visuals transformer, for feature extractor. And this is our deep embedding clustering, and here how we calculate our T distributions and how we calculate the target distribution, how it assigns the high competence assignment, which is calculated by using these equations. And this is our attention based method to select the most representative features. And this is our graph aggregations, how we calculate the equations. We calculate the similarity between the node by using the cosine similarity, and then we use the global optimist functions that assign either 1 or 0, and this is how we aggregate, how we generated our graph. And then this is our graph attention network, and here each layer we assigned the weighted and the updated weighted between the node I to j, and then finally we use the global mean followings to aggregate the features. And this is our experimental results. So here we use the two datasets. The one is the fetch statistics dataset and it's the breakage dataset. In the fetch statistics dataset, this is the As tomorrow's endoscopic images, here are total number of the patients 991, and the each image is 300 into 300 pixels. On the other hand, we use the Breakage dataset. There are two classes, either benign or the malignant. And this is our performance indexing. We use the blue, macro, raw, and these are our parameter settings. We use the total 4 LLM model for caption generation, and here is our hyperparameter meter settings. So now we will explain our quantitative results. First time we explain for a classification for a Breakage dataset. So here we see our loss curve. The curve loss gradually decreases, which indicates that our model can learn the more better way. And this is our quantitative result for caption generation. Here are our 4 LLM models. This is also same. The loss curve gradually decreases. That means the model generalized. So our research question, our main aim is to answer these 4 questions. We design the 4 questions and then we answer these questions. The first one, does the graph-based MLI performs the better than the SOTA MLI for microscopic classification? Does the spatial relation information, so the patches, impact the performance of the model? And do the LLM performs the better than the LSTM or traditional transformer models? Do in-domain LLM performs the better than the generalized LLM models? Here are our total 4 questions. Now we'll explain these questions. The first one is our quantitative results for classifications. We compared our results with the 4 famous MLI method. We can see that our method performs better than the other method. That means the graph-based aggregations performs better than the normal attention-based MLI method. Cover our question number 1, as well as the spatial relation affect the accuracy of the models. This covered our questions number 2. For better understandings, we also use TSNEB feature visualization. We can see that the red circles determine the malignant and the benign, and the blue color determine the benign. Yeah, there are some misclassifications. And this is our quantitative result for caption generations. Here are all the methods use the fetch-stick dataset. We can see that our proposed method performs the better than the all other SOTA methods. So that will cover our question 3, our question 4. So we can see that the LLM models performs the better than the transformer-based models. The here author 2, they also use the LLM models, but in our case, the in-domain LLM models performs the better than the generalized LLM model. That will cover our question number 4. And here we can see our quality results. Here we randomly selected the 2 whole slide images, and then the GNN-ViTCap model effectively generate the captions which are more closely to the ground truth captions. And this is our conclusion. So here we proposed a novel GNN-ViTCap process to classify the whole slide images as benign or malignant. And the GNN-ViT model is based on visual feature extractor and the clustering and the graph neural networks. And we apply the attention-based determining clustering for remove the redundancy and the graph neural network used to aggregate the image features. And finally, we compared our proposed model with the SOTA process, and the experiment shows that the model can be used for medical professionals with accurate whole slide image captioning. So our future plans, we can perform the better parameter differential fine-tuning for LLM models, or for clustering, we can use the adaptive clustering for improvement of GNN-ViT architectures. And this is our summary appearance. Thank you.